So today we'll discuss one of the important topic of CSIR-NET syllabus, and these are the diversity index calculation. So now, first of all, the what is biodiversity? Biodiversity is the different varieties of life forms in the earth. Now, biodiversity actually depends on and, depends on different factors like first is species richness, species richness. What do you mean by species richness? Species richness means presence of different types of species, right? Different types of species means, suppose in habitat A have species A, species B, species C, species D, species E. But in case of habitat B, here is species A, species B, species D, species E, and species F and G. So more number of species is in the present in the habitat B. So habitat B is the species richness is high. So species richness high means biodiversity is high. Now coming to second point. Second point is the species evenness. Now what do you mean by species evenness? The species evenness means the whether the species are equal number distributed in equally or not, right? That means if the species evenness high, then also biodiversity is also high. Suppose in case of community 1 species A is number is 10, species B 9, species C 11, species D 12, and species E 10. Now community 2, look at and community 2, species A 10, species B 15, species C 20, species D 5, and species E 8. Now look at that. In case of here, there is a in case of, see the number 10, 9, 11, 12, and 10, almost there is a range of 1 and 2, but here is you can see that 10, 15 says 5 difference, 25 difference, and in 5, so 15 difference. So there's a large number of difference, so evenness is less in community 2 and species evenness high in community 1. So one of the community have higher species evenness. Which of the now you have to find out from here which community have higher species evenness. So you can see that the community 1 have higher species evenness than community 2. So community 1 has higher biodiversity than community 2, right? Now coming to species third point is the species diversity. So taxonomically or phylogenetically different species in an area or habitat. Now you can you can see that the area, these are area 1, area 1, area 2, area 1, area 2, and area 3. Now here you can look at that crow number, different crow, peacock, and parrot. Now you can see that crow in area 1 is 4, peacock is 1, parrot is 1. But in area 2, crow number 2, peacock 2, parrot 2. In area 3, crow number 2, grasshopper 2, rabbit 2. Now here you can see that the species richness of the species in all the three area is 3, because there are 3 different types of species are there. So species richness is 3. Now species evenness, you can see that here is a 4 is there, but in case of area 2 it's almost equal, and area 2 the number area 3 also number similar. Now in case of a species evenness is equal in area 2 and area 3, you can see that. Now when the species evenness also equal, and then we have to go into the genetic diversity. You can see that in area 2 and area 3, now crow, peacock, and parrot, they are all belongs to the birds group. It's like, and but in case of here you can see that crow and grasshopper is the insect, and it's a arthropod, and rabbit is mammals. So there is a higher genetic diversity or taxonomical diversity is there. That is the area, that's why area 3 is the highest biodiversity. So when species richness is similar, species evenness also similar, then we have to go into the genetic diversity of the area. If the genetic diversity is genetically more diverse, then there should be, or taxonomically more diverse, then its species as a diversity also would be high. So diversity high in area 3. Now coming to ecosystem diversity, different types of ecosystem, that is no role of genetics here. Comparisons of two ecosystem is the ecosystem diversity. Now we'll come to that, the diversity index calculation. So actually the different ecologists and those are work ecological scientists basically use the diversity index. The now different types of diversity index or calculations are there for the measurements of diversity in a particular area, but depending on the questions asked by the scientist or ecologist, and different types of diversity index are used. Now here major two diversity types of diversity index we'll discuss. Another is the one is the Simpson diversity index, another is the Shannon-Weiner diversity index. Now what do we mean by in diversity index? First, the diversity index is a major mathematical measures of species diversity in a community, in a community. Now coming to Simpson index. What is the Simpson diversity index? Simpson diversity index measures the probability that two individuals randomly selected from a sample will belong to same species or same category or other than species. It is actually measures of dominancy. So Simpson diversity index is the D formula is the D equal to summation of small n by capital N the square. Summation of small n by capital N the square. Now what is small n denoted? Small n is the total number of organisms of a particular species. Small n actually is the total number of a particular total number of organisms of a particular species is the small n, and the capital N is the total number of organism of all species is the capital N. So here is the small n. So formula is summation of small n by capital N the square. Summation of small n by capital N the square. So D is the diversity index. Now here you can see that what is small n denoted. Small n is the total number of organisms of a particular species. Now what is capital N? Capital N is the total number of organism of all species, right? Now coming to Shannon-Weiner diversity index. Shannon-Weiner diversity index is this formula is H equal to minus summation of pi into ln pi. So H dash equal to minus summation of pi into ln pi. So Shannon-Weiner diversity index is here. See, you can see that Shannon-Weiner diversity index account for both abundance and evenness of the species present. Now, what is PI? PI actually relative abundance of each species calculated as the proportions of individual of a given species to the total number of individuals in in the community, and capital N is the total number of individuals, right? Total number of individuals is the capital A, and small n is the number of individuals of a species. So formula is H equal to minus summation of pi into ln pi. Now we will see how to calculate different problem. This problem came in CSIR-NET exam. So you can see that the question is following table shows the number of individual of 5 tree species in a community. Now species A is 50, species B 20, species C 20, species D 5, species E 5. Now based on above, you have to calculate the Simpson diversity index of the community. Now how you will calculate? So first write the number and number species A B C D E. Now you can see that the number that is small n is the number given, like in here is the 50, 20, 20, 5, 5, right? Now we have written that all number what is given in the question paper. Now small n by capital N. What is capital N here? Capital N is 100. Capital N, if you add, so 50, 20, 70, 20, 90, and 10, that is 100. Total number of species n equal to 100. So small n by capital N, small n by capital N, so 50, 50 by 100, that is 0.5. Now 20 by 100, 20 by 100, that is 0.2, and here also 20 by 100, 0.2. Now we can see that 0.5 by 100, so 0.05, 5 by 100, so 0.05. Now we can see that from small n by capital N the square. So 0.5 we can write it in also, is that 1 by 2, that is half the square, so it will come 1 by 4. Now 1 by 4 we can write as a 0.25, right? So 1 by 5 the square, you can calculate in that way, and you will get and add up the small n by capital N the square. So you will get D, that is diversity index 0.0335. Now you can look at the data here. The option is option 2 is the correct answer, 0.335. Now, coming to here, the now coming to the second question, which you have to calculate the Shannon-Weiner Diversity Index calculation. Now please look at the problem. The following table shows the number of individual of species found in two communities. Now community 1 and C1 and C2, these are two communities are given, C1 and C2, and species A, B, C, D. Now C1 community 1, that is 20 A species are there. Community 2, it's a is 80. Now 25 5, 25 0, 25 10. Now here also the value is given for that, for this a log value, because you have to use the log value. So whenever log value is given, you know that you must have to put into Shannon-Weiner diversity index, right? Shannon-Weiner diversity index is, the formula is minus summation of pi into ln pi, right? Minus summation of pi into ln pi. Now come into species, write the data, whatever data is given in the question paper. So write first and then put the data. So in small n is 25 25 25. Now you can see that in community 1, this is the community 1, the 25 all are given 25. Now for community 1 you can calculate the small n pi. Pi is the small n by capital N. Pi is the small n by capital N. So total capital N is 100 here. Capital N equal to 100. So 25 by 100, 0.25. In that way, it's 0.25 all. So 0.25, the value of my LNPI. LNPI value, you can see that the value is given LNPI for 0.25, 0.25, that is minus 0 minus 1.4. So we have just write that minus 1.4 here, minus 1.4. So pi into lnpi, so you have to calculate pi into lnpi. So in that way you will get, here is the total, that h, you have to calculate the h equal to minus summation of pi into ln pi. That means here you can see that the minus minus summation of minus 1.4, that means it's a getting 1.4 for community 1. The Shannon-Weiner diversity index is 1.4. Now we come into community 2. Community 2, the species A, B, C, D is there. Now here is that 80 5 5 10. Now see, in same way you can calculate pi, you can calculate lnpi, you can calculate pi into lnpi. So minus summation of, the formula is minus summation of pi into lnpi, so you will get 0.69. So in case of here, you can see that here one species is dominant, here a species is dominant, so biodiversity is low in community 2, right? Now we will see another is the type of communities is calculation for biodiversity index calculation, that is called community similarity. Community similarity. In case of community similarity, so here you have to calculate it, calculating the community similarities, right? So what do community similarity means? So calculating community similarities calculating community similarities. That is, what is community similarities? So what the communities have in common in terms of species, have in common in terms of species, helps us determine if we are comparing comparing apples to apples and to oranges to oranges. Now, so here the one of the important that is used is the Sorensen coefficient. So community similarity index, the formula is given by the Sorensen coefficient. So Sorensen coefficient, the equation of Sorensen coefficient Sorensen coefficient or CC, Sorensen coefficient CC equal to 2C by S1 plus S2, 2C by S1 plus S2. What is 2C? 2C by S1 plus S2. So where C is the number of C is the number of species, the two communities have in common, and S1 is the total number of species in community 1, and S2 is the total number of species in community 2. So now you can look at the examples for calculation. Now Sorensen coefficient from value Sorensen coefficient. So first look at the question paper. So first question we'll see. Question is, there are 20 species found in community 1 and 25 in community 2. 25 in community 2. Between them, between them, they have 5 species in common. Species in common. So you have to now calculate. So calculate the Sorensen coefficient. Sorenson's coefficient. So here the answer will be the Sorensen coefficient. Sorensen coefficient or CC equal to, the formula is 2C by S1 plus S2. The species common, here you can see that between them have 5 common species. C is the common total number of species that is common in community 1 and community 2. So community 1 and community 2, the number of species common is 5. So 2 into 5 by S1 plus S2. S1 is the total number of species in community 1 and total number of species in community 2. So 20 and plus 25 in community 1, there are 20 species, and in community 2 you can see that in community 2 there is 25 in community 2. So here, so 10 by 45, so it will come 0.22, right? So Sorensen coefficient, Sorensen coefficient gives a value, gives a value between between 0 and 1. Between 0 and 1, the closer the value, closer the value, the closer the value is to 1, the more communities, the more the communities have in common. Now, so complete community overlap, complete community overlap, if the two communities have completely overlap or completely similar, complete community overlap, complete community overlap is, then the Sorensen value, Sorensen coefficient value equals to 1, equals to 1 in, for complete community dissimilarity, for complete, if it is a complete community dissimilar or the community is dissimilar, then the value will be for dissimilar, dissimilarity is equal to 0, equal to 0. So if the community coefficient, so Sorensen coefficient is 1, that means there is a complete, there more the communities have common, that means there is a complete overlap. If the dissimilar, it will be completely dissimilar, it will be 0, right? So according to Sorensen coefficient, these communities, in these communities do not have much overlap or similarities, do not have much overlap or similarities, right? Now we can calculate another calculation. So next problem. Now see the question. So there are 15 species found in community 1 and 25 in community 2, 25 in community 2. So now here you have to calculate the Sorensen coefficient. Now we know that the formula for Sorensen coefficient formula is, or CC, equal to 2C by S1 plus S2. Now what's the, you can look at the common, the question, number of species in common, that is here is the, there is 15 species found in community 1, 25 species in community 2, and between they may have 12 species in common. So 2 into 12 by total number of species in community 1, that is 15, and total number of species in community 2, that is 25. So 24 by 40. So 24 by 40 is the 0.6. Now according to Sorensen, according to Sorensen coefficient, these communities have a quite a bit similarity or overlap, bit of similarity, bit of overlap or similarity present. Now another is the important part is the in case of Simpson Diversity Index, we have previously we have seen the Simpson Diversity Index. Now in case of Simpson Diversity Index, that there is a value for Simpson Diversity Index. So if it is Simpson Diversity Index, Simpson Diversity Index, so index scale is there, index scale. Now index scale and level of diversity, level of diversity. If it is 1, that is, so Simpson's diversity index value is 1, that means no diversity. 0.9 means extremely low diversity, extremely low biodiversity. If the Simpson diversity index value 0.8, so very low diversity. Very low diversity. 0.7 low diversity. 0.7 low diversity. Now 0 point, if the value is 0 0.6, that is moderate low diversity, moderate low diversity. If 0 0.5, so moderate diversity. Moderate diversity. If 0 0.4, then moderate to high diversity. If 0 0.3, high diversity. If 0 0.2, then very high diversity. Very high diversity. If 0 0.1, that is extremely high biodiversity. Extremely high biodiversity. If it is 0, the Simpson Diversity Index, so infinite diversity. So in that way, actually, scientists or ecologists calculate the diversity index. Now, which diversity index is useful depending on the questions that is asked by the ecologist, right? So depending on that, now today we have seen that these three diversity index calculation: Simpson Diversity Index, Shannon-Weiner Diversity Index, another is the Sorensen coefficient calculation. So hope that is helpful. Thank you.